Perfect.
All right. Then I will continue on here. You've got, so this is an introduction. This is an adult, and Mr. Splashy Pants. We've named them all here. That's because people can't resist naming them. I have a female named Sushi and one named Veronica, and one named Pandora, and one's Tinkerbell. And I've got one who's, I forget, starts with an R. Anyway, that's about all the females I've got that will lay eggs. And here's an example of some eggs. This is my student and the eggs. Can you see my pointer? My mouse? Okay, that's nice. So this is Tina, and she's helping me look after the salamanders and degel the eggs and cut them up or whatever I require. And she's around next year, which is nice. She's a math and biology student, and she's also taking anatomy and physiology next year. She wants to be a nurse. And you can see here's some close-up of eggs. This is a rect egg up here. And you can see the white yolky bottom and the top of the egg that has melanin in it. And these are freshly laid, so they haven't divided yet. And then I've got, this is an axolotl fry, freshly hatched. And this is the size of the egg in comparison. So they don't grow that much from the size of the egg to the hatchling. They have to rely on what's in the egg for nutrients. And I've got a series here, it'll come up. There it is, of some older pictures that I took of the egg starting to divide and then dividing further and further, and the cells becoming smaller and smaller and smaller until you get to about this stage, and then they tend to stay about the same size. And then this stage is interesting here as well. This is the brain here, and that's the spinal column. This literally rolls up to become brain and spinal column with the neural plate inside here. And this is the neural crest cells on the outside. And here's neural tube closure. And to meet all the animal regulations, I am not supposed to experiment on them past the neural tube closure stage. But I imagine I could get permission to take their photograph past neural tube closure, but I haven't broached that yet. I haven't asked. And I have this slide here. Developmental time depends on temperature. So that's why the stages are numbered. Like there starts at 2 here and goes up to 20 at neural tube closure. And it shows the number of hours at the varying temperatures. So if I had a very warm room at 23 degrees Celsius, I think it only takes 40 hours for neural tube closure to happen. So the device that I've got to take pictures of my embryos needs to operate continuously for at least 40 hours. And here's another picture of this brain and spinal column neural tube closure happening. And I've tried to enlarge this so you could see the cells. I think the cells 30 micrometers? Is that what you'd guess? Anybody? Are you still there? No? Have I lost you?
No, it's still there.
No?
They're
Just
still
muted.
there. Okay. Is 30 micrometers a good approximate size for these cells?
I usually put it at 15 to 25, but
Oh, okay.
you can measure it.
Yeah, the ones out here seem to be a little bit bigger. The ones inside are smaller. At least I'm fairly sure they are. I took a look at them earlier. And here's your theory. There's a wave of contraction, cell contraction that goes across the surface of these eggs. And we are fairly sure that this is a cell differentiation wave, and that the top part here, which is covered by this top part here, becomes the neural plate and eventually rolls up to become the brain and spinal column, like I had in the previous picture. And here's some theory about how that might happen. Here's a progression of this wave, and what happens is a microfilament ring on the apical side of the cell contracts, and you get the microtubule involvement, and basically the physical signal gets right down into the nucleus. And this is a picture of what might happen. So you got some sort of a force, and the stress is propagated through the cell and into the nucleus. Here, oops, sure, just move that around. And I managed to get a picture of it once. I'm hoping for more pictures of these. And there's a cell contraction here and here on the surface of a developing egg. Okay, it's about page 10 to 11. And of course, this happens in more than one type of animal. So this is somebody's picture of what happens to a Drosophila eye. And then we've got more apical constriction in sea urchin, and Xenopus, and Drosophila, and in chick. And because you're into C. elegans, I included this slide in the next slide. I guess I'm not sure of all of the mechanisms here, but it looked like kind of a nice overview. But they left out some important links, it said, between contractile actomycin networks and cell-to-cell adhesion proteins. And they're omitted in this diagram, so I don't know if it's helpful or not. And then here, there's an apical constriction happening in C. elegans. Somebody's diagram of it. So this is a before, and this is after. Anyway, so, and this is some of the significance of, well, a cell's substrate. If you have an undifferentiated cell and you put it on a stiff substrate, it tends to turn into bone. If you put it on a viscous substrate, it turns into cartilage. And if you have it on a very soft medium, it turns into an adipose cell, fat cell. And I've heard that if you have it on a liquid medium, it turns into blood. And there's lots of evidence out there for this type of transition. And then this is an example of the shear force and pressure pressure on endothelial cells. And this is without the shear force and pressure. And then the bottom one is with this shear force and pressure. And I've met people who say, oh yeah, if you're going to study the cells vessel walls, you need to clamp them off so that they're under pressure, or they'll undergo apoptosis and die, and you won't get any data. And here was an interesting experiment. I hope you can see that. If you have nothing happening to these precursor nerve cells, you only get a bit of development. You can kind of see a light, some green in that picture. If you stretch it, you get some nerve development. And if you add retinoic acid, you get nerve development, but you get more nerve development if you have stretch. And what they did was rhythmically stretch a rubber substrate, I think, under these precursor nerve cells. Anyway, it was statistically significant. So what I'm trying to do is look at all sides of the spherical embryo as it develops and look at stress and strain on the surface. So that's, it can be difficult because they're 2 millimeters in diameter and they have a lot of yolk, so you can't use a confocal microscope. And they come with a gel coat that sticks to anything, and they're kind of gooey. They're mostly water, composed of water, so they're gooey. Anyway, this is what I wanted to use, and it's over $70,000. So, needless to say, I didn't get that grant. I tried. But here, Dick, I hope you forgive me for this. I wanted to call it Dr. Richard Gordon's flipping idea, but this is the pictures that he took while turning an egg over.
Susan, that's a rather flippant remark.
Flipping remark?
Flippant.
Okay. Anyway, what happens is these are bottom heavy. So if you turn them upside down, then they right themselves. They turn themselves back the way they were. And so you can take a picture of them as they're turning themselves the right way up and get a picture of all sides of the embryo. So I devised this thing. This is my flipping microscope stage. And what it does, it flips with this part here. I'm just going to ruin my slides here. And it rotates with this motor here. So it'll rotate this way and then flip. And they'll rotate and then flip with this motor here. So this one here is a very fast motor. It's the fastest servo motor I could find. And then I had a student who was very ingenious at being able to program an Arduino to make it flip, because it wanted to check where it was every single time it moved, which slowed it down. But he got it to flip fast. So that was really great. I don't have the programming included in these slides, but if you wanted to see them, I can show you. Anyway, this was a machine just locally here in Winkler by Leon Bowman and General Metal, and they do a very good job. They're a good machine shop. So anyway, these are rollers that hold the wheel in place here, and this is on another servo motor a tiny nano motor, they called it. Yeah, it's a nano high-tech servo. And this is the entire setup. I've got a stand here that this is a magnification stack. It's an LM scope, and it's from Austria. And then you focus it here, and I've got a Nikon D7200 on top. I tried my Nikon 7100 and it didn't work. It wasn't fast enough. But the D7200 has low enough light capabilities so that it was able to properly take pictures. This is a light here. I've got side lighting for this as well as a ring light. It seems to need the side lighting, but the side lighting I was able to go to Walmart and get some nice LED lights. So that only cost $25 as opposed to, sorry, just a minute, as opposed to ring lights that can cost $500. I splurged. This ring light does cost $500, but it had angled lights and it was more durable than some of the other ones I had. Anyway, it makes nice even lighting. Now we'll get on to this. To my right here I have motor, and then these are the Arduinos. I've got two Arduinos. One runs the motors and one runs the camera. And this is just a start switch. And then there's a relay that attaches to the camera. This is a remote for the camera, and yeah, we stripped it and stuck it onto a relay. And we'll see if this works. Here is the stage, and I'm going to see if it'll play for me. There we go. And there's a embryo on top there. I don't know if you saw it. There it is, and it's being flipped. I haven't got it inside. I stuck it to the top of this. Like I said, the gel sticks to anything, especially rocks, so it didn't have any trouble sticking. I plan to put it inside here and then put a cover on it so it doesn't dry out, because they dry out very quickly.
That will be without the jelly, right?
Yeah, I can do it with or without the jelly. If the jelly is clear and I can see through it, then I could leave it in the jelly. Okay. But if it's giving me a bad time, if there's bits of junk in it, then I'll have to put it in the methyl cellulose. And I'm looking into getting the oxygen permeable membrane to put over top of the hole here. And then I can just fill it with salt water, Hofrader's solution, and hopefully the egg will like it in there. At least it'll have oxygen from both ends. So what am I trying to do after I get the images of these eggs, which I haven't done. I have a few of some dead eggs. I finally got my apparatus to work about the time the eggs were disintegrating. So I do have some pictures of a dead egg flipping, besides the one you saw. But I didn't get to see development. So I'll hope my salamanders will get busy and lay, maybe starting in November. I'll see what I can do. They just don't like to lay eggs in my classroom because it's too warm. Anyway, I've got a chiller coming, so I should be able to keep their water cool. And I've got the local museum. It's a fossil discovery center kind of interested in displaying some of the animals, and I think it's a better environment for them. So I'm going to try to persuade them to take on, like, say, half of the animals for now and see if they might be persuaded to start a breeding colony. I don't know. I'll have to see. They're kind of reluctant to say they're going to permanently take care of them. Anyway, back onto this slide here. If you're looking down a microscope at an image, you can't really tell what's going on physically with that embryo unless you know their density, viscosity, elasticity, their volume, and even the interior blastocele cavity volume and pressure of the blastocele, and basically where the yolk and the nucleus placements are in these eggs. You kind of need some other parameters, some physical parameters to go with your images. So I've managed to get the density out of them, with sort of a rough idea of the density. Let's see, standard volume measurement and weighing of these 2 millimeter eggs is difficult. So I used a density gradient column, and that seems to have worked, although it is on the edge of not working. The bottom there says challenges, small sample size, and living tissue. And so the sample sizes cannot be any thinner than 0.1 millimeters for my density gradient. And then for viscoelasticity, there's all kinds of ideas, and they're difficult. So I'll just start. There's the density. I made a sucrose gradient, and I bought a bunch of density floats. The density floats are very expensive. The stupid things are around $200 each, just for a little wee tiny plastic ball. But I have enough that I can take some fairly accurate measurements. And here is my density column with a whole bunch of eggs in it. I threw in whole eggs, I threw in chopped up eggs, I threw in rotting eggs, and they all collected in kind of a stripe here. So the estimate is that they're between 1.5 grams per milliliter and 2.5 grams per milliliter density. And they don't float, they tend to fall to the bottom of a water column. And I tried this to cut them up, and it's good for mashing them. And I've got one, yeah, that's fine for the density gradient. I was able to get the density of mashed eggs. And I've got one with a single wire, and I can cut them in half and find the difference between the top black portion of the egg, the animal portion of the egg, and the vegetal portion of the egg, which is the bottom. And I didn't get to do this due to circumstances this year, but I'm hoping that will happen next year. And so future density experiments that I want to do, get a statistically significant number of eggs and measure the top and the bottom of the egg to see the difference in density, and perhaps measure the neural plate compared to the rest of the egg. And I'd like to measure the properties of the blastocele fluid. Now, that brings me to this slide. These are all blastocele stage eggs. They develop a cavity. This is the outside. This is stage 9 egg. And they develop a cavity in here. And this is an MRI of the cavity. And this is the cavity when it was sliced. So you can see there's some distortion here when these eggs are sliced. However, I went ahead and I am doing some slicing experiments to see if I can stain the eggs. So this is what I'd like to see. I'd like to be able to see where the yolk is and the nucleus in these cells. And this is what we got so far. And the fellow that's doing it, Andre from the Department of Biology at the University of Manitoba, says these are extremely difficult. He tried them out in paraffin and tried slicing them, and he said they were like, they just went to dust, a lot of them. They just didn't work. But he's trying them with resin, like you would for an electron microscope, and hopefully I'll have a few more slides in a week or two when he's finished with that. And then he was going to stain them, well, just like they are here with the fast green and the safranin. And then he was going to try staining for the nucleus as well. Now I'm hopping back here to the MRI. You can see the blastocele, and you can see how it changes here. This is 11.7 Tesla coil MRI that was used originally to, the apparatus was originally used to measure a grain. But I'm going to try optical coherence tomography. It's less hard on living tissue than the MRI, that high a Tesla MRI. And so you can view these eggs actually through their gel. And you can also use them if we get it going properly. We should be able to do optical coherence elastography and see if we can get some measurements of viscoelasticity that way. Right now, this is the setup. And you have a swept source infrared light source. What was it? About 1300 nanometers. And you put it through an interferometer. And apparently a swept source, this is a key component of the swept source, this 3x3 switch here. And this is it's kind of a setup of the one in Toronto that we used for our trial run. And then I've got another diagram which might be easier to follow. You have your laser or your diode source, and it goes into a mirror, and it will. This is a reference arm here through a mirror, and then this is your sample arm that goes through here. I think that's it. No, I don't know. Anyway, you've got a reference and a sample arm, and this is showing how you can actually get a measure of some physical qualities of your sample by squeezing it. I don't know if this is making any sense. If you want to look up interferometers, that's basically how this works. Or somebody has described it as ultrasound using infrared light. And here's basically where it lies in usefulness. Confocal microscopy doesn't do this intermediate range here, and for salamander eggs, confocal microscopy is useless because of the light scattering properties of the yolk. When the entire egg has yolk. And then this is ultrasound, and ultrasound is too big because my eggs are 2 millimeters, so the optical coherence tomography should work for them. Maybe not quite at cellular level like I would like, but they keep improving the optical coherence tomography technique, so I'm hoping. This is the initial pictures I got using the system at the University of Manitoba in Dr. Sharif's lab, and it just shows the gel and then the outside of the egg, and it doesn't show anything inside. That's because this is a single wavelength source, and so we decided, yes, we should try this. This is a swept source. So it's 1310 nanometers plus or minus 55 nanometer wavelength light source. And here you can see the gel coat. You can see a water layer there, and the vitelline membrane or the start of the egg, and a blastocele cavity. And Dr. Sharif has just managed to build a optical coherence tomography system so that instead of part of the egg, which is kind of, this belongs up here, I can get whole pictures of it, so I can see how that blastocele grows. So I think that's a worthwhile study. And other people have done this as well. This is an 850 nanometer. I think this is also swept source optical coherence tomography, but this is a Xenopus Ache. And it shows why I'm looking at axolotl egg rather than Xenopus. These are smaller eggs, and they have a cell layer on top of this layer, this is the neural plate layer here, and then there's a layer of tissue over top of it, so you visually cannot see the neural plate development with Xenopus, or at least not as readily. If you had this refined, maybe you could. And I've got this. I put two images over top of each other, so this is the MRI with the blastocele cavity, and then this is the section that we did as a trial with the Toronto Imaging of using optical coherence tomography, and you can see they match. They actually, this arc actually matches this MRI. I thought that was quite neat. So this is actually the beginning of the neural plate in here, in this area. And the optical coherence tomography is superior to the MRI, partly because it's less expensive, but also because it looks like it's going to be less pixelated, so they have a higher resolution. And it also only takes a half a minute to get this image. Actually, it's shorter with the new optical coherence tomography in Dr. Sharif's lab. I believe it's going to be less than 0.5 seconds to get that image, and with the MRI it was 3.25 minutes. And the 11.7 Tesla coil caused defects in the embryo, probably because the microtubules become aligned in such a high magnetic field. So hopefully the optical coherence tomography, works so that I can take multiple images and it won't damage the eggs. Okay, here's the next technique I tried at the University of Manitoba. This is a diagram of an atomic force microscope and a picture of the real thing. But the problem was my eggs dried out. They dried out like within a half an hour of putting them in this machine, so that didn't work very well. So I went to a biophysical conference just recently, and they had this lovely atomic force microscope for liquid samples. But it couldn't acquire a signal because, I don't know if you can see it, but the yolky eggs decided to make the entire water sample milky, so the laser couldn't get a track on the microscope tip. So that didn't work.
The egg disintegrate?
It disintegrated, yeah, enough that it blocked the laser signal.
Oh, okay, okay.
Yeah, he hopes he's going to sell one of these to Manitoba, University of Manitoba, maybe. If he does, then I can take another crack at it, maybe try to glue the salamander bits down. I'm not sure.
How much does that thing cost?
I'm sure it's in line with this other one here. Costs, I don't know, a couple $100,000 maybe.
Oh, yeah.
Kind of. Maybe it's cheaper. Maybe it's only like 70, like the microscope that I want. Yeah, my budget doesn't account for these things. So actually I need a chamber that just keeps things moist and not wet and not dry. So I don't know if this would work, but it's called a closed perfusion chamber. I'm not sure how much that would cost either. At least $3,000 to get the chamber. And I don't need it heated, but they also come with heating quite often. Oh, and this is sort of out of order, but this is looking at viscoelasticity too. And it's an air puff tonometer by Dr. Fleury. Is he from Paris, France?
Yes.
Yes, okay. And he's developed a system where you can get a puff of air onto your sample and then measure how well it springs back. I think this could be done with the optical coherence tomography setup and a puff of air. We could try it. The other idea I had for measuring viscoelasticity was using microbeads. You have to be careful. The probe, if it gets small enough, just measures the cytoplasm and not the tissue that you're looking at, so I need a larger microbead. And I mentioned it a little later, or maybe I've mentioned it just down here somewhere. I met someone at the photonics conference, and she had optical tweezers, and she was quite willing to try to measure the elasticity of some sample eggs for me with her system once she got set up. She was from Laval, Quebec. So we'll see. Maybe I can do a partnership with her and I can get some measurements.
Has anybody tried a sonic system? Bounce sound off the membrane?
Bounce sound off of it?
Yeah.
I might be able to try that, and you could use the optical coherence tomography and a sound wave. That's an idea too. Okay, well, Do you have any more information on that type of system?
No, I just crossed my mind that maybe it seems like everything else has been tried. Maybe that would. There's probably somebody tried it. I have no idea. It just popped in my head.
Okay. All right. Well, thank you. What I thought of trying was maybe something like this. There's I have had some correspondence with these people, and they're looking. They finally got their blood viscosity meter launched and on the market. So now they're looking for other uses for it, and microreology might be the way to go for other, like if they want to measure phlegm or something from that people cough up, then I'm all for it, because that's exactly the consistency of my eggs. So I'm hoping. Anyway, this is a MEMS device, and the University of Manitoba, I talked to them about it, and they thought maybe they could possibly make something like this too as a student project. I don't know. There's some good news. I don't know if it's here. No. There's a microreology device coming to, I think it's chemistry or physics at the University of Manitoba, and they could probably just take an egg and stuff it through the device, and they might be able to get me some microreology measurements. So I'm hoping for that at the moment. Okay, other projects. I also met someone at the photonics conference that thought I should try measuring pressure using optical fibers. So I'm going to try to launch that project. And then I saw an article about someone, I think, aiming blue light at neural precursor cells and having them change into nerves. And so that got me interested in how light interacts with microtubules. There is a photoelectric effect with microtubules. Although if you look up photoelectric effect microtubules, you won't get any hits on Google. But if you look at light and microtubules, you get lots and lots of information. Yeah, and there's the University of Manitoba acquiring a microreometer. I think it might be chemistry. Anyway, I've got the contact. And then I want to make this nanotech device for measuring viscosity. So a nanotech beam or an inkjet printer viscosity measurement. And then there's the optical tweezers that I mentioned before. So that's basically all my slides. Kind of ran through them. I'm also interested in this. It's a neural crest. This is where you have the brain and the spinal column, like the stage, what was it, 18 or 16? This was an experiment that was done in the 1950s, and they put vital dye on the neural crest areas, and you can see where it spread throughout the animal. I thought that was very interesting. So you could follow some of that. And also something that's physical are the pharyngeal pouches that occur when the animal curls around the beating heart when it starts to develop. And there's, it's just like folds. If you curled something up, you get folds. And there's pressures involved in that. So the study of that would be interesting as well. Anyway, that's all my slides. So I will Do I minimize that? Yeah. Okay, there we go. Can you see me now? Yeah. Okay. Stop. There we go. Okay.
Thank you very much.
I have my apparatus here if anybody wants to see it. There you go. Just keep it all together.
So how fast is that flipper working now? Fast.
Oh, you saw it in action.
Yeah, it looks like half a second or so.
Yeah, that's the fastest servo I could get. And it seemed to work for the animals in their own gel.
Have you timed it? And how fast does it turn upside down?
No, I haven't timed it. You want to time it? I'll find the PowerPoint slide and time it. No.
You might need a strobe.
Oh, I can time it, yeah. I should time it to write this up. I have to write it up. I've been given a couple of weeks here to write all of this up, so I haven't done it yet. It was nice to do the PowerPoint because now I've kind of got a start on that. Yeah, I need the timer. Here, I'll write it down as a note. Timer, time flipper. Or time flipping? There we go, time flipping. See if I know what I was talking about. Anyway, yeah, that was the fastest servo I could find. Kind of expensive, but it did work. If I can increase the speed if I increase the voltage, but I kept it at the 5 volts that came off of the Arduino, just for simplicity's sake. But if I powered it with 7 volts, then it would be faster.
Okay, but it's probably fast enough.
Yeah, I think it's fast enough.
Yeah. Okay. Okay.
So it's really fascinating, that part about how stem cells differentiate based on the fibers inside. What are the myosin fibers? Like how stretchy they are on the surrounding surfaces. It was amazing.
Yeah, well, it's just the substrate. For mesenchymal cells, the substrate seems to make a difference. For the nerve cells, stretching seems to work. Or stretch and then what? So there's pressure in the blastocell and it's stretching them, and then maybe the pressure diminishes and then they turn into a nerve cell. I don't know if that's part of it, but it certainly looks like the cell cramps, the differentiation wave cell cramps, help the cells turn into a neural plate. But I'm going to take full images of this and we're going to check it out.
Hey, Susan, do you have ultrasound available? Or at least a sonicator?
Somebody has ultrasound.
Or just a sonicator, not an imager, just a sonicator.
You can get one from a French company.
Okay, what I'm thinking is one issue that was of concern, and you might check the literature on human embryos, was, women have ultrasound tests to look for developing embryos and fetuses. And I don't know if this was ever resolved, but I remember one concern was that the ultrasound might affect the embryo.
Yeah, it's possible.
Now, if you think of the blastocell, for example, as a bubble, it has a certain dimension and it's in a chamber of a certain elasticity. That means it would probably have a resonant frequency. Okay? So
Yeah.
one might disrupt development by hitting that resonant frequency.
Okay, poor eggs. They're getting.
Yeah. But the thing is, you could then have some concern about iatrogenic induced defects. Okay? Okay? So, you know what I'm saying?
Yeah.
Probing the mechanics of an embryo, it might be through ultrasound resonance.
Okay.
Okay, it's just occurred to me. I remember that old concern about ultrasound and embryos, and I don't know if it was ever resolved medically.
Yeah, they say it's rare. I remember being ultrasounded when pregnant, and I felt warm, and I was very unhappy because if you have a raise in temperature, it can cause heat shock proteins and damage to the spinal column, and I was very annoyed.
Yeah, well, there's an increase
So
in
why don't you come here? Oh, Natalie wants to say something.
there's a lot of controversy about ultrasound. Most of the studies seem to be that it is entirely safe and that there's nothing there. But there are a couple of studies that, one in Norway in particular I can think of, where there was an increase in the percentage of the population that's left-handed in populations where everybody gets ultrasounds. And while there's usually a percentage of the population that is left-handed in any case, and it doesn't mean anything, when you have an increase in the percentage, it usually means small, subtle forms of brain damage. So left-handedness by itself is not diagnostic of brain damage, but an increase in left-handedness and having left hands, being left-handed in addition to that is. And part of that is probably due to heating. Part of that is if you have an ultrasound at a critical point of development, but the ultrasounds that are being used now today do a lot less heating than the ones that were used 30 years ago. So anyway, I personally would not have an ultrasound unless there was an absolute medical reason for it. In that case, the benefits would outweigh the risk. But a lot of people have ultrasounds for what I regard as spurious reasons. And I think the worst ones are the ones where they do ultrasounds to get portraits that are like happy pictures of baby, find out the gender of the baby, and to make a beautiful series of pictures that the parents can put together as like baby's first picture. And for profit as well. I'm very much against those.
Yeah,
Anyway.
for medical reason. So
Yeah.
mine was for medical reasons. So okay,
Yeah.
but I still was not impressed.
Okay, so it's interesting. So it could represent another probe of the physics, because looking at music, without a resonance effect, then you might be picking up a critical physical parameter.
Yeah, another physical parameter that I'm going to have to check with the flipping microscope is the fact that I'm flipping those eggs, and vibration has an effect on a developing Xenopus. I know I've got a paper about that, and also gravity has effects on developing embryos. So much so that in microgravity, I don't believe mammals can reproduce.
Has anyone tried it yet?
Yes.
Oh, it has been.
They sent rats up.
Oh,
When do rats not reproduce?
and the rats didn't reproduce.
No, that just is not right.
Did the rats get past the first step of reproduction?
Yes, they did. They checked it.
Okay. Okay.
Yeah, that was quite interesting. You just,
Amazing.
it's probably microtubule intensive or something.
Oh, okay. Wild. So it's not obvious whether, well, see, yeah, I guess repeated turning over may or may not have an effect. But we don't know until you get some eggs and just keep them in the flipper for a long time and see if they develop normally.
That's okay. I'm going to have a control. They gave me permission to buy another LM scope end, and so I can have one that's stationary and just take a picture of the top of the egg as it develops, and then the other one I can just I can flip it.
Okay.
Yeah, so I've got a control now. I didn't mention that in my outline, and they gave it to me anyway. That was nice of them.
Well, good.
Yeah, so that's about all, I guess. I fight with my salamanders all the time. They're fussy and they won't eat their food. They make their water extremely dirty. They don't like it hot. They want it at 18 degrees, and then they don't want to lay eggs. It's just, yeah. I have pets.
Okay. Okay. Well, we got to get you some more females then.
Yes.
Well, I was talking about those fibers again inside the. It seems like the strains within there, cause some stresses within the nucleus, and then it affects the genetic expression, right?
Yes. Is that
So my analogy was it's a party favor. You just pull the string and it goes off.
Oh, okay. So that was like.
It's a joke. Okay.
Okay. But it's more like one of these very fancy fireworks where it sets off one and then it sets off another, so you get a very complex fireworks. Okay? That we sort of nested party devices.
Did you want the references?
Yeah, how about sending us the slides?
Okay, I could send you the slides. Maybe I'll clean it up a little bit and send you the slides. Or maybe it doesn't matter. It's kind of rough. I didn't spend a lot of time at it, but just collected them from here and there.
Okay, Bradley, can you put the slides in a place we can all see them? Okay, so I'm sending them to Bradley then.
Okay, yeah, I can do that.
Mhm.